Okay. Protein-protein interactions, terminology. I briefly mentioned this before. So we're talking about physical transient protein-protein interactions. What I show here is the complex of GP120, the glycoprotein 120 in, in HIV. So that's the green part here. And you see this glycoprotein binding to a protein called CD4 and to two antibodies. If she disturbs you, tell me. Two antibodies that are trying to detect it. So the entire complex you see here, that's a three-dimensional structure. And ultimately, for me, a physical protein-protein interaction would be the connection between these two. Because they go on and off. They are contacting at some point in time, and they are contacting only for some point in time. Another point of contact would be this, the red one here. Now, ultimately, when you look at pathways, so A activates B, activates C, activates D, then you could imagine, or you could argue that all of these are interacting. They are somehow, in some sense, interacting, but the correct term here really is they are associated. So the physical interaction happens only between A and B, between B and C, and between D and C and D, but not between A and D. So they are not connecting physically. Okay. The same way here, the antibody 2 and the GP120 are not in connection. One way experimentally in which you get a high throughput view of many proteins interacting is a system called so-called yeast-2 hybrid. It's called yeast because it's done in yeast, and you essentially have these two flags, the AD and the BD here. You tag two proteins X and Y. You want to know whether X and Y bind. The moment that A, D, and BD come together, something happens that emits light. And they only come together when X and Y bind. That's the idea. That's sort of a high throughput system in which you do it. So the simplest way in which we can infer that two proteins bind is simply by homology-based inference. So it's the story that I showed you before for localization and many other examples. So assume we have protein A and B, and we know experimentally that they interact. Assume further that we have another protein A prime and B prime, or another pair, A prime and B prime, and they have some similarity. So A prime has some similarity to A, and B prime has some similarity to B. Now the question is, do A prime and B prime interact? Okay? I know A and B do. And these other two are sort of related to A and B. So can I then infer that in fact A prime and B prime interact? And that is essentially called homology-based inference. What this one shows here for H values that are very high, so 70 is about 100% sequence identity. So in this curve, that is number of residues aligned versus percentage sequence identity, the blue is the H val 0, plus values are above, so plus 70 is somewhere up here. Essentially here for some part of the spectrum is corresponding to almost 100% sequence identity. And you see that still, let's not talk about what this factor is, but what you do see is that the higher this number is, the higher the red number somehow, right? Okay, there is an extreme error bar here. You cannot really fit a line, or you can fit many lines into this one here. The line that you could fit could be something like this. And in some sense you could fit a bunch of straight lines. It could actually be a fairly straight line, right? To me the data it appears as if the data supports more this idea that there's some growth. So that the line, in fact, that fits best to these red arrow bars here, again, the green ones here are the averages of the distribution, red are the error bars. And again, so also you see that it goes up and down, so most likely this is a problem in the small data set. But most likely, I would argue that the line that best fits is not like this, but it's something like that. If it is something like that, then what you see is that for higher sequence similarity, this homology-based inference works better. Okay? This is what we expect. This is what we have seen much better with more data, more clearly for localization. That's what we have seen for protein structure and many other features. Okay, now what's the issue here? Why is that so poor in some sense? Why is the line so part of the problem really is I don't have enough data to do it. Let's get back to this word homology. Homology again is sort of coined historically by something on the level of an organism, or at least on the level of an organ. So there's a homology between the fin of a whale and the hand of human. That is what you call homology. So the whale has the same kind of expenditure or some organ, and that corresponds in human to something else, but it's homolog. It does something similar. For dog, bird, you see it does sort of the same thing. For whale, it does something slightly different. Now, in the genome evolution, so there are two different words, and again, I do not expect that you will remember them, but I still want to introduce them. They're so-called orthologs and they're paralogs. And the idea of an ortholog is, so when there's a copy of the genome and there's a new organism coming, and the new organism, so when there's a split into two species, then both species essentially have the same genome, right? Because at some point they were together and then they split. And then these two split species here essentially have very, very, very similar genomes, okay? And very similar genomes means also for every single protein, essentially they have a corresponding protein. Because just before the split, at some point, they were the same species, all right? So that means they have a corresponding protein for everything. So for every protein in organism in species 1, there's something corresponding in species 2, okay? And that is essentially what is called ortholog. So the idea is these are the corresponding proteins that do the corresponding things, that do the same thing. It's the machinery, the same parts of the machinery. It's the four wheels in a Porsche and a VW bus or something, or in a van. Now, the paralogs is this idea that now, since you may get additional genes, with these additional genes that you get, you could do something new, okay? Because you have the orthologs already. You have the four wheels, the car can drive. Now, if I threw another two wheels at it, you could do something else with it. So you could create a truck and have a six-wheeler or something like that. And the idea then of a paralog is that duplication events lead to the creation of new proteins that do new things, okay? Now that means that the new thing here may look very similar to the old one, but actually is free to evolve a new function. That's the idea, right? And the way it happens, so into species, it comes through duplication, and then ultimately when we are going down the tree, the trick becomes to recognize what of those things we have. So if we have a protein between two species, is that the ortholog? Or is it the paralog? Is that something that does actually the same thing? Or is it something that sort of has evolved a new function? Maybe the other species has evolved into something that really is very different. Okay? Well, whale and human have arguably some differences, not only in size. Have you ever heard of jumping genes? So the idea of jumping gene is that you put a gene on a table, and then you stand and wait, and you see it jump. April 1st? April Fool? Does something like that happen?
Yes.
Yes, because it's biology, or what? It was actually first discovered in corn in 43. Here's a sea slug. So in fact, it is when you look at corn, you see the wild corn has different colors, and they actually come from from jumping genes. Okay, so this is also what the first many years ago they had the first of genetic experiment in Germany, and what they did is they had a large field of daffodils that were white, and then one red one in there, and they wanted to see whether that would jump, and others would turn red, and there was a lot of an uproar against it, a political uproar against gene experiments. Unfortunately, this experiment didn't work because it was summer, it was too warm, and all the daffodils died. So that was the end of that experiment. But jumping genes exist. And they are fairly frequent in the plant world. but here's a sea slug. You see a lot of those. Now, horizontal gene transfer, yes?
That the gene jumps means that it jumps within its own DNA stream, or does it mean a duplication? It jumps over?
It jumps to the next plant.
Ah, okay. So it just embeds itself in someone else's DNA.
Yes.
How?
Where's the jump coming from? Actually, I don't know. So most likely there's a spring attached to it. So it cannot jump by itself. Most likely it's attached to some part that lets it jump. So in bacteria you have bacteria actually shoot ammunition at each other. You can call them arrows or something like that. And they shoot fairly long distances. I assume it's some other similar mechanism. I actually don't know. Never asked myself that question. For bacteria, the way I have always imagined it is more that they are floating around. There is a lot of horizontal gene transfer. This is the same thing. So two bacteria swim next to each other, and one gets the genes from the other. So most likely this is the example of the arrows, because bacteria constantly communicate. But actually, eukaryotic cells also communicate. So I do not even know whether the jumping genes are only extracellular proteins. We'll look that up. We'll try to get an answer for you if it's known. Anyway, so let me skip this. Can we predict protein-protein interactions from sequence? And the first way in which I tried to do this was the following idea. I take a part from protein 1, so shown here is 1, 2, 3, 4, 5, 6, 7, 8, 9 consecutive amino acids, and a part, 9 consecutive amino acids from protein 2. And I have some overlap region here. So these 3 from 1 and the 3 from 2 overlap in this region. And I feed this into a machine learning device. So initially I took a neural network here, and I tried to predict that so this is a consecutive window of 9 consecutive residues, 9 here, that the central ones are in contact. Okay? As simple as that. You believe this works? I did it, right? So it must work. No. So this was, I failed often in my life, but this was the most formidable failure. This absolutely doesn't work. Doesn't mean there are no publications of people who claim that it works, but it absolutely doesn't. And the problem really is there are way too many false positives. Meaning that now what you have to do is you have to slide this through both proteins. And there are too many things that possibly could bind. So when two proteins bind, and now I say there are too many regions that could possibly bind, what could prevent them in real life from binding? So in the computer I can slide all possible 9 MERs past each other, right? Why can some possibly not bind? Well, it's already written here. So the answer why some are not written, here's the first way of reducing it. Because proteins essentially bind on the surface, right? So if these two regions that are slide are somewhere inside of the protein, they will never see each other in real life nature, in cells, right? In a computer they will. And maybe those regions are perfectly bonding to each other. But since in the protein they're not on the surface, that will not work. So this I can address by simply predicting the surface residues. This is what we talked about in subcellular localization. So instead of sliding this window through the entire protein, I only slide it as long as I have at least 1 residue or 2 residues in this window that are on the surface, or whatever mechanism I have. Now, if I sort of, rule of thumb, the protein is roughly an elongated sphere. So now, what do you call the surface? Let's just say half of all the residues are on the surface, okay? If half of the residues, and again, it's depending on your definition of what exactly you mean by surface, but that is one reasonable definition. Now, if I took this 1/2, then this mind you, would reduce the false positives by a factor of 4, right? Because now I don't have to scan the entire thing anymore. I only scan 1/4 against 1/4. And that ultimately is a 4-fold reduction of false positives. So this is a simple trick by which I already can sort of 4-fold go down in my false positives. The way I do this, so this is again, there's the core, there's the surface somehow, this is the surface region, the dark green here is the core, and I try to predict this with machine learning. Now, the next issue is, what if I could predict residues that are involved in protein-protein interactions? And I'm going to go into this one, so this will be for the remainder of today. Let me just continue here. If I could do that, I could reduce even further the false positives. And then what I would do is I would look at the prediction of the residues that are saturated internally. I could even reduce more. Then I look at things that are in the same subcellular localization, and I say they interact only if they are in the same subcellular localization. It's not always true. So there are, of course, interactions between nuclear and cytoplasmic proteins, because they have to meet each other. But I will ignore them. I will just say anything that is off the diagonal I will not allow. And in a situation where I have 1, 2, 3, 4, 5, 6 different states, and say they were equally populated, this reduces my false positives by a factor of 36. Right? So again, I will lose some true positives, but most of the stuff that I will throw away, in fact, are false positives. And that is done through this method. You may remember that this method, log tree, in fact, even has 18 states. So again, if they, so 18 square, so I reduce by 18 by the power of 2, whatever that number is. So the next project would have been to predict residues in protein substrate interfaces. And again, if a protein binds a substrate, it cannot bind another protein. So I can exclude those. Then I have to predict domains. I have to improve alignments. And then I have to put all of this together. And then I can predict protein-protein interactions. And I will, in fact, talk about that maybe beginning on Thursday, maybe early next year. Let's begin with the data collection. So the data collection is, ultimately, I take all of these 3-dimensional structures, and I look at all the data points, and I want to predict whether or not I have this red type, whether I have this red type here, or the blue type, or I have internal interactions. So I would want to see whether different interactions look different. Why is that important? Well, if I want to predict interactions in the way I create my data, the question is, when 2 residues bind internally in a protein, should I distinguish that from 2 residues binding between 2 proteins? So what I see here in red, should I distinguish that from what I see here, for instance, in green? Why is that relevant? Yeah?
Well, if the goal is to predict protein-to-protein interactions, then you don't want to have protein within one protein interaction.
Okay, so now when you think about this, you have this stretched out amino acid chain. You put it into a solvent, it adopts a unique 3-dimensional structure, so it folds, right? And it folds by the fact that some amino acids attract each other, sort of, right? So you have parts of the sequence that come together. There are forces that attract them. Now, the forces that attract them inside, why should they be different from the forces that attract them between 2 different proteins? Well, you're right. It could be that the biophysics of the story, while you're right, this is what I want to predict, but the biophysics of the story could be the same thing. It could be that, going back to the example that I had before, it could be that the only reason why some of these fragments that I had 9 MERS do not interact is because they are not seeing each other, they are not on the surface. But if I would sort of pull this out from the, and now I have to sort of really make a ball, a protein ball here. So what I mean is that these 2 strings bind out here, if I pull them out and had them on the surface in two different proteins, maybe the same thing that binds internally would bind between two proteins too, right? Now that is relevant for me because in my protein database of these internal interactions, I have a lot. I have a huge data set. In fact, this is so far in biology the best problem for which you can really do deep learning. You have millions and millions and millions of data points, right? While of these types of interactions, I get into a million, but just. And not millions, not many millions. Certainly not like internal contacts. So, okay, let's first see whether they differ. What would you do? My question is, I'm going to first ask, so we have internal, we have these external between two different proteins here in red, and then I have permanent external. So permanent external is an example here in the yellow, but it's cut off here. You have two different domains of the same protein. I call them permanent because the same protein is always together. These two domains will always be together, right? And those are three different types of interfaces. And my question is, do they differ? How would you answer that? And you would answer it like any other question. These days you would answer. You would do the same thing. What do you do for any other question that you don't know? Yes, you would try that. Now, what you will find, so at least what we found when we did this, is we found publications that said they differ, and we found publications that said they don't. So what do you do next? How do you handle that?
Flip a coin.
Do you do that? when you read two types of articles, so in this particular case, in fact, in our case, we had more than two publications for each of these points. Would you then flip a coin?
Probably not.
What would you do?
You don't experiment yourself.
But before you do that, you do something else. I believe you will do something else.
Check which one you trust more.
That's exactly what you will do. And trust has to do with, for instance, is it a better journal? That is one way. Is it a better conference? Is it something that has a higher reputation? Another thing that you will look at, do I know these people? And this, in fact, is one of the reasons why in science, networking is very important, because knowledge of people is something that in these moments sometimes makes a difference. In either of those two ways. Knowing may mean I know I cannot trust that person. Knowing may mean I know I can trust them, right? And then comes the other story. You look up where they come from. So you have two groups from some small university in the Midwest, or you have two publications from Harvard. If the two Harvard Harvard and Yale or something like that, if Harvard and Yale fall on the side they differ, and these two others fall on the side they don't, you tend to, whether it's right or not. Most likely I would argue that if you counted thousands of these examples, most of the time you would be right in doing something like that. In this particular case, none of these algorithms worked. We knew these people, but the ones we trusted were on both sides of the fence. And then we come exactly to what you said, you do it yourself. Okay. So here we have, unfortunately, do it yourself does not always work that fast. So now the Google story, that was an afternoon. And then the student and I discussed that. That was all still part of the same afternoon. Getting those data, that was many, many months later. And many months later, so those are the 20 amino acids. What you see here, 0 means they occur as often as expected. So you compare it simply the frequency to all the frequency in your database, okay? And then there are these three different types. So minus means less than expected, plus means more than expected. So internal is green. That is sort of the transient protein-protein interaction in red. And sort of the permanent internal ones are blue. And I'd like to play this game where every single one of you in the room makes a statement about what they see. Okay? And this is exactly what you do in your results section. This is how you discuss when you have a figure in your thesis or in a publication, that's essentially what you do. You write what you see. Who wants to go first? One small statement, not a big summary, just the result, no interpretation, just what do you see? Who goes first? Everyone has to pick a pie. Whoever picks first has the simplest thing to say. Yeah?
Well, the amino acid W, for example, doesn't appear as often as expected in protein-protein interaction.
So this is always very negative.
In general, I don't really see a pattern that fits for all of them.
Ah, that's too fast. Begin with a simple results. So something like that. So begin with one residue or one thing that you see. Not in general for all. This is not what you begin with. Come, there are so many things we can talk about. What is not a valid statement is there are 3 different colors, because I explained them already. Not laughing. So in the figure caption you have to say that there are 3 colors and what they mean. Which again is one of the common problems in most theses, that students just don't write what the axes mean, or most people in the talks don't say what the axes mean or what the colors mean. Say something else. Yeah?
Maybe in each group, like from R to, we can see that the protein-protein interaction is more frequent than the others.
So you mean here, or in each of these? For each amino acid, you mean?
The lower. Yes.
But this is minus. This is less often than expected. This is more often than expected. So let me turn this around. Here the red is lowest, here the red is lowest, here the red is lowest, here. So there are at least 5 of the 20 where the red is lowest. Now this is the next statement would be already sort of interpretation of this, would be the discussion section. But 5 of 20 is actually somehow, if it would be completely random, then you would sort of expect the number higher than 5, right? So at least the cases where it's very clear. Maybe now you sort of count a little bit more detail and you see, wait a minute, how many did you count? 1, 2, 3, 4, 5. So yeah, it's really true. So if it would be completely random, so then that is not a random event. Should be 3 out of 27. Well, maybe close to 7. Next observation.
Well, I think my understanding of this graph is not really great, but I think...
Is that the statement?
I think the bar is going down, but that's my pre-statement. Now my statement is that just looking at the lengths of the bar is going down on the negative side seems to be larger than the ones going on the positive side.
Yes. So this is again, you're getting into the general statement. For somebody who doesn't understand the figure, that is already a big jump, right? So when you don't understand the figure, you begin by one point. Also, when there's something in your thesis or something in what you find that you don't understand, begin to sort of look at outliers. Begin to look at some extreme points and see whether there is some sense you can build around that, right? And then begin to sort of integrate it into a bigger picture, general, so what you do, Ole, is already a jump. But it is actually true. So what does it mean? Ultimately, it means that the kind of usage is always less often than expected. That means the 3 interface types, all are not random. And that again is another observation. Whether it's negative or plus, there is nothing here that is 0. And not consistently 0, right? So the way interfaces are formed is not the way protein sequences are built. They are a special subset. Okay? This is already one clear result. More simple things.
I just got a question regarding the diagram. Is the order for the amino acids on the horizontal axis important, or are they grouped like that, or is it?
Yeah.
Just random?
So no, it's not random. So it's not random. So these are two positively charged, these are two negatively charged. We get into sort of the hydrophobic residues. There is something special about those here, and there's something special about those.
So
they are somehow grouped at what you call properties. But in some sense you also see that this idea of properties is not completely supported by the data. So if we group by properties, then, and we would say with these properties we can explain the protein, then clearly this is another thing you see. It doesn't work, right? There's no clear, consistent reaction. So there are only two positively charged residues. This one is very, very clear, R and K, and they behave completely differently. There are only two negatively charged residues. Okay, here you can argue they somehow, actually they somehow appear to behave similarly. Now, the hydrophobic residues here, that's the group of hydrophobics, they are not quite similar. In particular, the I and the L, people always say, are completely exchangeable. So not for interfaces. The part of the red is similar here, but the blue, well, there are differences, right? Anyway, now, Madame, now I'm doing exactly what you're supposed to do. I'm taking your time away, yeah?
The G is quite similar between the changing and
That's an interesting observation, yes. So that gives many people the opportunity to say something else. Because essentially, Jessica, what you say by saying they are similar, you pick one where something special happens. And the special thing is they are similar, which we can of course turn around. Almost always these bars differ, right? That's another implicit way in which that you said. You see something else?
I think the most extreme correlations are with chain-chain and protein-protein versus internal. For example, with R, they both chain-chain and protein-protein go very far in the negative.
So you say that blue and red are similar here, blue and red are similar here, and green is different, blue and red. So blue and red are somehow more similar to each other than they are from green.
Yes, but not always, but more often.
Well, that's a statement that is certainly, so now what you would, in fact, in order to make that statement in the table in the results section of your thesis, you would have simply to count, right? Is it true? So I don't know whether, not always, but most of the time whether you count it, whether it's 11, but you should do that when you interpret figures, right? Most means 11, right? It is true. But the interesting observation is we tend to make these statements, look at a graph like that and say, oh, most of the time. But that's science, right? You really count until 11 at least, and then you can say most of the time, right? Anything else?
I just have a question. How much did your data differ from the ones that you saw in the articles?
That is an unfortunate complication in there, completely. And so our argument why we couldn't trust those papers is because they looked at very different ways of doing it. And some, in fact, had used all the data and had not really distinguished, had a different label for what we call red than what we had. And some had carefully chosen the same kind of labels, but they had used only 20 proteins. So we used 1,000 proteins. So we needed, and in fact that is why I said it took several months. I believe this point took a year. So this took, getting the data was a long, long step in this particular case. Anyway, train doing this. Train doing this with your own results. Train doing this with people who talk. Just see things. In particular, find outliers. So look at extreme cases such as this one here, where all three completely are very, very negative. Or look at cases where, in fact, they point in different directions. So examples, well, where was the, or examples where this one here is clearly the high, or whatever. So look at some features and see what you can make up an idea that you call a hypothesis, and see how much the data that you see supports that hypothesis. Okay, now what we do know is they differ. We see that, right? These bars differ. The question is, are these differences meaningful? They all differ from zero. They all differ from each other. Now, does that mean that the biophysics of these interactions is different? How would you answer that question? Whether they are meaningful, those differences. Yeah?
Maybe define a threshold and then say if the difference is like this big.
This is exactly what you're supposed to do. The problem is, what it implies is that you have an intuition for what is a meaningful threshold. Now, I've been in this business for a long time, but I don't have an intuition for amino acid composition or for amino acid differences, for these bars. I don't have an intuition. So I cannot define that threshold, not by intuition. Well, you can go around and find somebody who has more intuition than I. Good. But I challenge you, this will take a while to find that person. So what can you do before you find that person? Again, Google doesn't help. What can you do? If you don't find a person, how could you sort of find a simple detour? Any idea?
Put this data in a machine learning device.
Yes, that's a big step. That is that would be sort of completely nailing it. If you can predict it, then they are different, right? This is very clear. And I will get back to that. But isn't there something simpler? For most publications, you do something else. You should have grilled me already that there was something in the graph that I didn't show you. What is missing in this graph? So if you get out of this lecture, this is the one thing that I hope you will learn. To jump at people who show you graphs like that, yeah?
Pretty much the reliability of these?
The error, yes. The error is missing, right? So there are big differences here, but do they matter? And the first way, how do you compile an error? Remember? Statistical significance. That's the minimal statement you have to make. Here, you would apply a simple chi-square test and simply ask, are these two distributions, do they come from different distributions or from the same distribution? Now, the problem with chi-square is it doesn't work if you have very small data sets. Chi-square is applied in medicine, and in medicine typically large data sets are 10 patients, and that is where very often 10 patients is a large data set, and that is in fact a very, very small number, and chi-square does not really work. We don't have that problem. We have millions of proteins, or millions of points. Well, there's another bit of a problem here. That is, when the problem is very large, it also doesn't work. And in fact, this may exactly be where we are. What we have is the significance of these differences is 10 minus 300. And that was simply the precision of the computer used at the time. The Excel spreadsheet had a limit at 10 minus 300, below 10 minus 300. And you know, typically, if you have 10 minus 3 in biology, or 10 minus 5, you publish, end, right? Now, this particular student sort of had a strange idea. He asked, well, what if I put proteins that are called A or B in the first letter in one set, and proteins that are called C or D in another set? And again, I ask, do they differ? 10 minus 300. What if I looked at proteins that are published by one author and all proteins published by two authors? 10 minus 300. Then what if I had a random subset of proteins? Still close to it. Now this one fell a little bit, but we still came close to the 10 minus 300. And that then got to the issue, why didn't it work? So then you go out and you talk to statisticians, and they ask you, well, is your data independent? Well, proteins evolved from proteins, so nope. You see, you cannot apply any statistical test for molecular biology. This is, of course, not the kind of statement with which you can easily live, because the entire molecular biology is full of statistics, right? While the chi-square or the statistical independence assumption is clearly not valid, there are many tools out there that work, although sequences are violated, biosequences are violating that. Now, this is the point at which we came up with another idea. And the idea here, we call it find self test, to do something like statistical significance. So we have 3 data sets. We pick out, say, 1,000 samples from each data set. Put them into these pots here. And then we pick another set, another 1,000 from the first. And then we ask, with this 1,000 here, which of these 1,000 is that, in terms of information content, by adjacent channel information, most similar to? Most similar to this, most similar to that, most similar to that. Right? And if you repeat this now, this random picking, 1,000 times, and if it always finds this one, then it's significant. I show you here the results for 1,000 picks. Now I don't only have 3 different types of interfaces, I have 6 different types of interfaces. The green is still the internal, and this one, the red one here, is the transient protein-protein interactions. Now the first thing you observe in 1,000 picks, the diagonal is always highest. So we can clearly distinguish between these 6 different types of interfaces. The next thing you see is that when you have confusions, so the largest confusion here is the hetero-obligomer confused with domain-domain. Okay, now hetero-obligomer, those are, obligomers are things that are always together, that I've never seen not to be connected to each other. So these are essentially 2 different parts of the same protein that are together, right? Domain-domain is essentially the same thing. It's a connection between 2 different parts of the same protein, except for, in this case here, I call it domain. In this case, I call it different part of the protein. And that is sort of a mistake maybe in the way I label my data set. So this confusion makes a lot of sense. Let's put it like that, okay? But ultimately, this is the part that I care for here. So almost 900 of the 1,000 identify the class of external protein-protein interactions, again, at least for 1,000. If it works for 1,000, then it clearly states that the biophysics of these interfaces is different. That much is clear. And by the way, for random subsets, this one here is completely given random results for the other tests that I showed you before. And now, since on an average I can see that they differ, the next question is, can I predict? Okay? Now comes the point of predicting these binding sites. The biophysics differs, can I predict? Now the way I do that, I take the protein database, I find 130,000 about at the moment. I find all the ones that bind, and then I extract the interactions. I'm not going to rush through this. I'm going to take the time. So the question is, how do I extract the interactions? What we do know is we have to label them. But the real question is, so these are 2 different chains here, or 2 different proteins. There used to be the glycoprotein 120 from HIV. That's the CD4 in blue here. And what I mean here is there are 2 residues in the green and the blue that have some proximity. Now what I talked so far about were points that already were collected after I made the decision. The question is, at which threshold do I say green and red touch each other? Okay, and this is the same type of problem. You go into Google, you ask what's the typical threshold for the binding, you find different answers, and then you sort of measure it yourself. what we did is smaller than 6.5 angstrom. Now, we have a data set and we can apply machine learning. One thing that I want to show is whether the cutoff is a 6 angstrom or 4 angstrom in yellow. What I show here is I pick 9 consecutive residues. And I ask, in these 9 consecutive residues, how many of these 9 are involved in the interface? And you see that for many cases, so when you look at the blue, for many cases, about 6 on average are involved in the interface. So the blue here goes with a peak around 6. Meaning that if you have any residue in an interface, in fact, typically you have more than one in the interface. This is not trivial. It could have been that protein interactions, so before we actually saw this graph, we didn't know. So enzyme activity, you have 3 parts here that come together. Okay, my fingers are not good. So these 3 parts here come together and they bind a substrate, right? That's the idea. And these 3 parts here are from far apart in sequence, they are far apart from each other, right? So now if I had, these are the 3 binding sites, if I now looked at the 9, there would only be 1 in each of those, because nothing else is binding in this one. So what I see here means that protein interfaces are sort of more elongated. And that was not necessarily clear. You see that if I make the decision of what I call an interface at 4 angstrom, so they have to be closer, the whole distribution, the blue moved to at least 1 or 2 residues. Meaning that if I require that they are closer to each other, I have fewer residues in the interface. That means they're a little more spiky in that sense. But the next issue is, how do I actually choose this 6 angstrom threshold? And that has to do with here's one issue in which I try to predict the flexibility of a protein. What you see in this particular graph, there are regions in this protein here where, so there are different models that I show here. One model is this light gray here, where you see dark, you see 50 models that come at the same point. It's a superposition of 50 different lines. And then you see regions where all the lines are somewhere else. And then you see regions where all the lines converge to the same points. And where they are different places, the protein is more flexible. Where they are at the same places, here, the protein is very rigid. Okay? So essentially what you see here is different time versions of that protein. Okay? And the most movement typically here where the red arrows are has to do with the function of the protein, and therefore in fact I want to predict these sort of flexibility regions. When I look at a large data set, the distribution of these so-called B value looks like that. The different colors here are different quality structures. In that direction, rigidity increases, so that's where all the lines before are the same. And in that direction, the flexibility increases, that's where you had this. So that side is here, and that side are points such as this one, okay? Rigid, flexible here. Now, if you wanted to predict, is the original data, if you wanted to predict the B value, and if you say took an SVM, you need a binary system, right? So you need a threshold. Where do you pick the threshold? Yeah?
Probably around minus 1.1 or something.
Minus
so
1 point.
There where the graph is pretty steep.
Somewhere here.
Yeah, around there.
Uh-huh. Okay, there was another point.
I would figure that the mode of this distribution.
At
the mode, so at the mean.
Here. Okay, so let's begin with the first statement. Your pick is by far the most common reaction. And the way I say that, you see where I'm going. I'm gonna shoot it down. But this is what most people in most publications exactly do. They pick the peak. Now the problem is the following. When you pick the peak of the distribution, you pick the point of highest value, right? Now imagine you at a peak, the peak, and imagine, here's your peak point, imagine there is some epsilon error. So you made an error in the measurement, right? So it's actually not that value, it's a little bit epsilon plus or an epsilon minus. Say epsilon is your experimental error, right? Your classification, because of this error, will go wrong, right? Now if you pick the max, you pick the point at which you're most sensitive to experimental errors. Because you picked exactly the point where any experimental error, no matter what it is, will have the highest impact. That's the problem with the peak. So if you have a sample set where you essentially have zero error, then that's a valid process. In biology, you don't. In fact, so I said that these three here are different quality structures, and already their peaks differ. So that's the minimal error that you have. In fact, the error is higher than that. So this is there are many things that speak for it, but the problem really is in machine learning, what you ultimately end up by classifying this is you increase the number of points for which you confuse the machine learning. And you choose the point that is optimal for messing up the machine learning. That's the problem with that. So let's go to this point here. The proposal was to take a point that is very steep. Now, any point is better than the max. So in that sense, this pick is a better one. So my question to you is, why this one? Why not this one? Sorry again?
I said, why not the other?
Can I wait with that for a moment? That would have been my next question. But the question here first, if you were to go to the left of the distribution and you go into this very steep part, why not go into a little bit less steep one? Because then again, the epsilon will affect you less. So let me begin this here. Can you answer yourself or anybody else? Why not going here?
Now the data is very unbalanced.
That is the least thing. Not only is it very imbalanced, actually if I get to pick this point, there's nothing left. That's the major problem. And ultimately that may be the reason why not go to the other side of the distribution. So the other side of the distribution also in terms of this error argument looks better because this is steeper. Right? So this is a little bit, the peak is not symmetric. This one is a little bit more flat. So why not go a little bit more on this side? Now, the story becomes, can we go here? And then we have the issue that there's no data left. So we have to pick a point somewhere. And we have to pick a point somewhere where we sort of have enough data. And from that point on, it is sort of arbitrary. We pick these two different points and call these two different modes and compare them.
Shouldn't the medium be somewhere around there too?
That I can't remember. No, I don't believe we ever looked at it. So I don't know. Could be. I don't believe so, though. Well, maybe. There's a substantial part here, yeah. Maybe. I don't know. So when you look at sequences again, so different types of B values here according to these thresholds, rigid, flexible is in red, you see they differ, much more so than the graphs that we saw before. In this particular case here, since this is all residues, you do not have them all differ from zero, they sort of sum to zero. Then that is sort of a prediction device that is for the non-strict mode. So the left more is sort of somehow similar to your initial pick on the other side of the curve, and this one here is a little bit further away. And you see you can sort of reach different levels of accuracy. In blue, the non-strict mode is actually higher. But this method at least allows you to predict in some cases proteins for which in the beta propeller where the functional site is rigid, and examples where the functional site is very flexible. So we predicted that this one is flexible, we predicted that this is rigid, and we predicted that both of those are functional. But at the time being it was a cover of a journal. So it works somehow. But ultimately, the problem is where to pick the point, and in some sense, we should do that here too. We should somehow see how the machine learning works, pick a good point in terms of the threshold. The 6.5 is one of those points. It's trying to be off the peak in a distribution, and now we can apply the machine learning. So the first question is, how do you choose the input features? So now I want to predict which type of interface I am in from sequence. I have my data. I want to try a machine learning device now. My next question is, what is my input? So in this particular case, everything we had so far doesn't work. This is not a problem where I look at the amino acid composition of the protein, because I want to know which residue is in an external interaction, right? So this must be a much more local feature. So far in the lecture we haven't had, I mean in this second round of the protein or the computational biology lecture, we haven't had an example for that. We had examples for that in the first round. Yeah?
Yeah, we do it like in secondary structure prediction, basically, so annotations per residue.
Yep. Let's go into the generic issue. When you do your thesis, this is the moment to walk around in your group and ask people. As many people, and bother them, keep bothering them. People you may not feel that they like you very much after that, but it's also true. So if you keep asking questions such as, where do I switch on the power for the projector? This is not making people happy. But if you ask people that sort of tap into the scientific intuition of the people in the group, that sometimes makes them proud. You just have to find the right way of doing this. And this is the kind of thing where you really need answers. And then we get to the same story that we had before. You hear different things and you don't know how to integrate it all. Well, let's just hope that you get enough input and then you can stumble over that problem. Okay, Konstantin made this analogy of secondary structure prediction. So we take a sequence of, in this particular case here, 13 consecutive residues, and we ask whether the central one here, a P, is in fact in a protein-protein interaction or not. So that's the hidden, that's the two output units, and each of these bubbles here is the 20 amino acids. It's actually 21, because we need a so-called spacer unit, and the spacer unit is because I'm now going to slide this window through the protein. And the moment that I slide this window through the protein, I want the first residue to be at some point in the center of this window. If the first residue is on the center of this window, before it is no residue left, right? So, and that is where the spacer unit is for. The 21st unit here is one that counts for non-amino acid. So end of protein. And you have the same thing if you shift the window through the other end, right? The last ones. Is that clear why we need 21, everybody? Anyways, there's a substantial part of the people in the room who have not attended last semester's lecture, right? No? Who has not been there in 1, 2, 3, 4, 5? So those 5, you got it? Okay. Now, here's another issue.
Maybe you can repeat it.
Okay. So I want to code for 13 consecutive residues. I'm not going to do 13 on the whiteboard. I'm going to do 3. So, and I'm not going to.
Is it a permanent marker?
Thank you. Yes, it is. So you can laugh about me afterwards again. That's the Christmas cheer. Let's just say I had a protein that has only two types of letters, A and B. So A, B, B, A. Say that would be the protein sequence, okay? Now, I want to look at windows of 3. Let me just get out the window of 3 here. Of these, it did happen. Of these ABBA guys. If I look, oh no, 3 is not ABBA, ABB. So this is ABB. Now, the way to code that would be, I have 1 unit for A and 1 unit for B. It's the A unit, that's the B unit. So this one here is on. Then I have 1 unit for A, 1 unit for B. Here's the B unit. 1 unit for A and 1 unit for B, the B unit. So this would be a 6, a vector of 6, coding for ABB. Okay? Now the problem with this, and again, this is the same as here, except for here is 13, there it's 3, and it's 20 amino acids, there is 2. Now, this would work, but what do I do if I want to code for A, B, A? So the first 3 residues. And if I in particular want, so the coding is always what state is the central residue in. The central residue in this window here is the B. If the central residue should be this A, so then the A unit has to be on, it's the B unit for this. And before it, there's an AB unit. But before the first residue, there's nothing. The first residue is the first residue. Right? So I cannot get away with a coding where I have only 2 units. I need another unit here. And that's the sort of not amino acid, or I call it spacer unit. So that's the A, B unit. And here's the spacer unit. Then A, B, spacer. A, B, spacer. And now I can have the A in the middle. The A in the middle will be, this is on, the spacer is on, and then comes a B. So this one, that's the spacer unit. This one is on. So in order to have the first residue be in the middle, I could do 2 things. I could say, well, my window is 3 residues long. I will never put the first one in the middle. So I will ignore all the windows before the first. And then I can still stay with 2 units. 2 times 3. But if I don't want to do that, if I want to really cover with a sliding window every single residue in the protein, then I need to introduce a new unit. This is why I have 21 here and 3 in this particular case. Okay, so I could, the reason of using 2 output windows, let's just say it turned out to usually work better instead of having only 1. And there are reasons for that, ultimately because you still have a lot of parameters and you can optimize that decision. Anyway, what I show here is 2 different decisions. So this is the output value. These are 2 different output values for this unit, 2 different output values for that unit for the same. This is one input vector and this is another input vector. In both cases, the decision would be that the central residue is in this unit, in P, in the protein-protein interaction. Because that number is higher, right? 0.9 is higher than 0.1 and 0.6 is higher than 0.4. In both cases, the decision is top unit. But here, the decision is much stronger than here. The difference between these values here is much higher, right? That's why it's a stronger decision. And we will use the strength of the decision here as a reliability index that we will see whether it correlates with some features. in this particular case, we use all kinds of additional input, evolutionary information, we use secondary structure prediction, we use solvent accessibility because we know that the surface composition is important, and ultimately the most important input feature is really profile, predicted 1D structure that includes solvent accessibility, membrane regions, and disorder, and some predicted aspects of function, in particular here B values. Now, let's see how well it does. So in order to see how well it does, of course we have to do cross-validation. So we have to sort of hide some proteins in our test set under the table. We have to take the PDB, protein-protein interaction parts, take a unique sequence, unique subset there out. We cannot do random split. We have to use every single case. We have to avoid overlap between training and cross-training and testing. And then we essentially can sort of plot it. Just using sequence information, this is already the entire graph. I cover some parts. This is the coverage. That means of all the residues that are to be predicted, how many did I get? So I have a data set of things that are in the red ones in external protein-protein interactions, right? How many did I get? And at some point here is 100%. And that is how accurate is my prediction. And you see, all I get here is the light blue, for some residues that are not even 10%, so this is 5% of the coverage, so less than 5%, less than 5% of what I could have had, but at least I have numbers of 60% or something like that. Is that good?
It's better than random. Random would be 50% if we assume uniform distribution and so on.
That is correct. So our distribution is not uniform. So in a two-state prediction, random is much higher than 50% because in fact it's about 10% of, I assume it's about 10% of the data that is in protein-protein interaction. 90% of the data sits in non-protein interaction. So random would be 90%. So this is way worse than random, but actually what I'm showing here is only positives. And for that, the 10% is random. Unfortunately, the random is not shown here. The next question is whether the strength here helps. And this is sort of correlated to the strength. Oh yes, the random comes here. So it's not quite 10%. This is one way of doing random. Now, before, this curve here was all on the left. That's the scale. So I'm less than 5% here. Just blew it up. The scale. It's the same plot as before. It still looks as dismal as the other one. But at least in some area here, for high reliability, you're clearly better than random. Random is down here. And here's another statement. For 1 in 10, at least, we get 94%. We get this may not be too bad. And now, since we know that protein interface residues are somehow clustering adjacent, we could apply that as a filter. So we could now say, okay, we're going to throw everything out where we have single peaks. So where in a window of 9, I have only 1 or 2. Again, I will throw things away that are right. But I will throw away a lot that are not right, simply because I know that on average, the blue is our definition here, on average, I expect something like 5, at least in the window. I can't remember what exactly the filter is that we applied. We said something about we need at least 3 in the window or something like that. And then we got to this one. This is still not very impressive, but now we are clearly away from random, and it looks sort of okay. Now, when we put in the alignment information, so alignment information means I'm not only putting in the window here from the protein, but I'm asking in a family of related proteins for which I know nothing other than their sequence, how often in that family do I see at that position a D? So this comes 100% D, this comes 100% P, this is 50-50 or 30, 60% and 40%, and those are the numbers I put in, right? That's all I change. Everything else is the same, and that's the dark blue. So now, in fact, I have it's not quite right, so the interaction sites here, the dark blue, also includes predicted 1D structure. This was sequence only, and this is sort of the jump you can get from using additional information. Okay? So ultimately, the jump from the light blue to the dark blue also has to do with how clever you are at choosing the right input information. That's the simplest thing you can do. What I showed you before, you take the window, and you take only the amino acids in that window. You already do better than random, but as you can see, you can do way better by putting in more information, or putting the right information in. So this is, again, what I show you here is the end of a long line of trying things out, trial and error in some sense. Now, again, clearly better than random. What would you say to something such as this one here? How would you explain that if you saw a data set? Okay, so this is the moment where you have to shout at me again, because there's something I don't show you. What could that be? Yeah, at least one person is smiling. Thank you.
Statistical.
Yeah. So this is clearly where we need error estimates. This cannot be. This is clearly an example where the data set is too small. This is typically what you see when you have some strange effect from the small data set. And you will see that the errors here, there's a peak in the error in that realm because I just don't have enough points. Okay, so there's another story. There are so-called hotspots, the protein-protein interaction hotspots. And the idea of protein-protein interaction hotspots, and I should have had a figure here, is, let me do it in a 2D way. So we have protein 1 interacting with protein 2 here. This is 1, and this is 2. And that's the interface. So the interface that I show here involves typically 20, 30 residues. Okay? A hotspot is operationally defined as maybe there are some of these interactions, so 30 residues are involved, but maybe there are 2 or 3 that carry most of this binding affinity or the binding energy, the free entropy or entropy of binding. And the way it's experimentally looked at is you change that residue to some other residue, alanine, and you knock out this binding. So there are cases where changing a single of these 40 residues will stop that binding from happening. Okay? But there's only typically 1 or 2 or 3, or there's only a few of these so-called hotspots. Now, there has been a debate on are hotspots real or not? And that debate is important because hotspots is exactly what you target when you do drug design. The ones that glue the most, they are the most interesting for designing drugs. If you want to stop this interaction here through a drug because that is relating to a disease, and if there would be one point in which you sort of can prevent it from happening, then that's the point where you design a drug for. Drugs are not big enough. They cannot cover the entire interface. So in the entire procedure of drug design, the assumption that there is a hotspot helps. Or is helping a lot in arguing. That's why this discussion, is there a hotspot or not, is so important. The people who argue against this idea of hotspots, they say, well, it's a gradual difference. So you have some of these interfaces, some of these pairs here that are stronger than others. But it's a function. There's nothing in this function. So we have here I show 20 amino acids. Let me just say 30, so that you don't confuse it with the 20 different amino acids. So 30 residues involved in the binding site, and that's the entire energy of binding. And maybe there is one that carries this much, and others that carry much less. And so if the people who argue that there are no hotspots, they argue there is no threshold in this curve that is such that you can say, okay, left of it are the hotspots. It's just a curve. And it's a little bit more to the left than to the right. That's the way this curve is defined. But there's nothing special about those hotspots. Okay, that is essentially the argument of the people who speak against it. And that is a debate that has been going on for 20 years, or more than 20 years in the field. Now, when we saw our data, first we asked, those few things up here, where we have a very good performance, is there anything special about them? Are they hotspots? In setting up this method, we never used the concept of hotspots, right? We just looked at the threshold, 6.5 angstrom, and trained. There was no training on energy. There was no training on some are more important than others. We just trained everything that we had above 6.5. We trained on everything that is entered into this curve here. So how would you test it? Whether your system sort of picks hotspot. Well, you cut off at some high reliability, and you get some data set of somebody who in fact measures hotspots. In this particular case here, the lab next to us, so we did that, still work from our Columbia time. The work next to us, the lab of Wayne Hendrickson, in fact, is the same that did the structure that I showed you before for the HIV protein, glycoprotein. And they, in fact, for this HIV glycoprotein binding to CD4, they showed that the red ones here are the strongest. They are the hotspots experimentally, okay? Again, our method never used the concept of hotspots. We simply looked at the most reliable ones. And here they are. So purple is predicted, red is observed. Here the whole thing is projected onto the structure. We did not use the structure. We did that from sequence. What you see is that sometimes here we have one over predicted, sometimes we have a red one that we miss here. So here's only one red one. We predict this one also. Here, this one here is what that we are missing. But overall, these two things are pretty similar. Overall, we actually predict a lot of these hotspots. Is that enough to publish? So you try one example, the one example works. What would you do next?
Try another one.
Yes. And that started, we started actually another year. The person who did that, worked on it for another year, and then ultimately came up with a larger data set that was also published later, in which at the time being, so again, coverage versus accuracy for a larger data set. I cannot remember how many proteins these were, 100, a few hundreds or something like that. He collected a larger data set. And then again, so what you see is for some, the prediction is really right, and in the worst case, it sort of looked like we get 60% of the hotspots. So the method really worked. And that was impressive, and we could publish that. But the real cool thing about it is this machine learning device answered a biophysically meaningful, important question. Hotspots exist, okay? There's no other way that I can predict them if I haven't learned it. I did not optimize on this data set. I never optimized on this concept of hotspots. I predicted hotspots without ever using the concept. And if I can predict it, then it exists. So this is an example I like a lot, because here machine learning, yes, we have a method that does something. But there's another aspect to it. The fact that you can do this means that maybe the function really is gradual. But there still is whatever that feature is. In machine learning, we cannot tease out what the feature is. But there is a feature that I can train with which I can distinguish between some left side of this and some right side of it. It may be arbitrary where you put the threshold. But ultimately, at the end of the day, there's a clear difference. And the story goes actually further. So in this particular case, Wayne Hendrickson, the one who had done the structure here, was on the thesis of Yanai, and that ended up in this thesis of Yanai. And in this thesis, he then asked him, well, this is what we published, but only one residue is real. Which one? So which one is your top? And Yanai got a very, very good grade because actually his method picked that one right. And he got even the first 3, but for that he then needed to use the structure. Anyway, so sometimes lucky coincidences help you. Okay, I believe at this point I'm gonna cut it and go leave the hub story here for Thursday. Yes.